Welcome to the open and reproducible analysis of light exposure and visual experience data online course, which very heavily focuses on one specific analysis package called LightLogR, which I am the main developer and creator of, but it all fits within this bigger framework of reproducible science, where we desperately need data to be available in a way that extends beyond the initial data collection efforts, because it becomes just more and more effortful to collect these data in a good fashion. So, we need smart ways how to collect, analyze, and publish these data. And so what I want to bring to you in the next 1.5 hours roughly is to get you up to speed from this jumble of data to the left to getting to a interpretable way of displaying your data, calculating and processing your data. So this will be really about the analysis side of things, even though obviously it is connected to many different aspects of data collection and so on. Before we get into some of the context, just a very brief statement about our funders. What we are doing today and the development of LightLogR would not be possible without the projects that are funding our efforts. So, huge shout out to our funders there. And so what we are doing in the lab is thinking about a more of a holistic framework about these data that we are collecting. I mean, we are collecting them for a reason, but usually they are very much useful beyond the initial data collection efforts. And I think the data sets that are just starting to be published as part of the Melidos project, and some of them you can see here, different data sets around the world. We are adding Costa Rica to the set just today, are really a great showcase why it is so good to have all these data at the fingertips, and all of these data are open and published. So you can download them today, you can work with them, do some analyses with them, compare them with your own data. And we really need to get there in order to get to a worldwide understanding of what light exposure, personal light exposure looks like. And it's not enough just to put a data availability statement in a paper saying that it is available under reasonable request. A very recent publication just coming out a few days ago called "Data is not available upon request" found that really in less than 1/5 of the cases where you find these kinds of statements, you actually get data. So I think it is really a very worthwhile effort to think about open and reachable, fair data in the first place. What am I talking about when I'm saying it gets so much more effortful or so much more work is involved in collecting these data? Depending on where you're coming from, your mileage may vary, but I'm coming from the non-visual effects of light in a circadian context in the research, and usually those were done with stationary measurements in the laboratory. You take a measurement, you connect this measurement to your protocol into a scenario, and basically that's it, and everything else you control for. And as we get to an understanding about what light exposure in the real world is like, collecting data with wearable devices, it gets more complex. And I'm not talking about, or not just talking about, file sizes here, because they are very much increased, but also in terms of complexity, because suddenly you're dealing with a time series of data, and those are to be analyzed very differently from those spot measurements. The complexity here is increased because there are very few standards out there on how you actually collect these data and where you collect them. So there is a large variety of devices out there in the markets. You don't really have a common set of output quantities that you find in every device file format. The metadata usually is very much limited. File formats vary widely. If you've ever dealt with time zones and timestamps, it can be really messy. You have to deal with gaps in your data, and there are just so many metrics out there in published literature, so it's hard to know which one to choose and how to apply them to your data set. And so the basic idea behind all of these efforts in the analysis package is that if we solve these topics in a bi-lab level, then we are wasting valuable research time, because we are going to spending efforts reinventing the wheel, doing the same functionalities, implementing them in our scripts, and they can be error prone. So that's the idea really behind LightLogR, which was created or started development roughly 2 years ago, but it has grown ever since. It is an analysis package for R statistical software. And the way we think about it is really as a one-stop shop for dealing with data from wearable devices. So we want to provide rich import functionalities that get you up to speed very quickly with the data you're collecting, that has rich set of functionality regarding validation and processing of data, so that it is ready to calculate metrics, to produce visualizations, and do the formal analysis, obviously. There is also a very important topic regarding metadata, so that we can more easily share data across different studies. And while we are not quite there yet, we are hard at the implementation of a metadata descriptor in LightLogR to make things even more powerful in the future. And LightLogR really is developed out in the open, so we are trying to do the best we can regarding transparent documentation, tutorials, robust unit tests to make sure that the package functionality is kept across certain development stages, and also regarding web tools, so you can access some of the functionalities without actually any programming knowledge. Today we will focus on the right half of this picture, so all about the functionality of LightLogR and how that factors into some of these thought processes of analyzing these types of data. LightLogR is very readily and easily available because it is hosted on CRAN, which is the central archive for packages in the R environment. So you can very easily install it and get going. And before we get into more of the documentation side of things, I think it is worth mentioning what LightLogR is and what it is not, just for you to have a little bit of expectation management going on. So we are seeing LightLogR as a feature-rich analysis package that is developed for time series data from variables, and it is a framework to prepare and transform your data in order to make the formal analysis. It is deterministic, so you are going to get the same outputs every time when you put the same data with the same commands in. And we are purposefully diverse about inputs and outputs. So we are trying to incorporate as many devices as we can, and we are trying to not limit you towards what your actual goal is. So, for example, what kind of light exposure metric you're choosing. But we would rather like to have a common interface for these functions. LightLogR is also very much opinionated, and it tries to complain whenever it can. What I mean with that is that because there are so many ways on how you can do something, you need a sensible default, and that means the opinionated part. So LightLogR will make suggestions that we feel are sensible, but that you can adjust and customize. And whenever there is something wrong going on, it tries to complain in a very verbose way so that you know what to do about it. So that's what LightLogR is in our opinion, but there are things that it is not, and the first of that is it is not feature complete. There are a lot of things that we would like to add, but just haven't gotten the time to do it or have found the right way to implement things, but we are working on those. It is also not a statistical method for inference. So the formal analysis, you have to work with the packages that that fit your scientific question hypothesis. LightLogR is there to prepare your data for it, but you need a different method for the actual inference because it is deterministic. It's obviously not probabilistic. And it is also not a substitute for good study design. So if you're collecting garbage data, LightLogR might be able to produce a nice plot or calculate metrics, but it doesn't really help you towards the bigger goal of your study. So you still have to make sure that the data you're collecting is actually meaningful. And obviously that also includes that LightLogR is not a replacement for theoretical understanding about these topics and critical thinking, because it will absolutely allow you to calculate nonsense if you don't know what you're doing. So reading the documentation is a very good idea in these cases. It's also not an imputation tool to guess missing data. I think that is more of a principled approach here, where we believe there are different ways how to deal with gaps in the data. If you absolutely have to impute missing data, then I think there are great packages out there that do that, and we are not trying to replicate those. So I mentioned documentation, and we are having a very rich documentation homepage. Later on in the tutorial and the webinar, Erina will put a link to the documentation in the chat, and you can have a look at this for yourself after the webinar. There are tutorials on there, function descriptions, so you can find all the good stuff there that you need. 2 or 3 sentences about the development of LightLogR. It is very much an open source project that is community driven. So LightLogR really lives in a GitHub repository that you can access, you can fork it, you can contribute code if you have an idea for a good function. If something doesn't work for you, you can submit an issue here, you can describe your problem or your feature request, and people have done so in the past. And you can contribute to making this package better tomorrow than it is today. So really, this is an effort that we hope is one that it's not just the few of us in the lab, but a bigger research community behind that. And we are using all the tools that are available in there, including things like unit tests to make sure the functions run as expected and give the output as expected. So over 800 unit tests at the moment. There is continuous integration and continuous deployment happening in the background, so every new feature gets to you as fast as can be. So one last thing before we go to the live section. LightLogR is developed with light exposure and light exposure variables in mind, but it is not limited to light. So if you are interested in other aspects and in ocular health, for example, there are a lot of different variables that are of interest. For example, distance, spectrum. In other contexts, it might be activity that is also recorded with your data. LightLogR has got you covered there. You just have to change the defaults because we are doing a beginner track today. We are more interested in the overall process, so we won't go deep into different modalities of variables. But if you're interested in that, there is a very nice tutorial out there that you can visit later on that goes specifically into things like distance and spectrum with much richer data sets than what we are looking today. So you can even look at distributions in space, so spatial distributions, in this case of distance, and with a little bit of tweaking, you can work with LightLogR in that regard. So with that, I would like to go over to the live portion of our webinar, and this is something you can join in. So if you have a setup with a second screen or a large display, then you can open the link in the chat that Erina is just putting there and follow along. And if you don't have that, don't worry, the link will stay active even after the webinar is finished. So you can go in there and within your own time discover all the different functionalities. But we are going through that one by one. And we have to choose our track, which is the beginner track. And there are 2 links here, one live and one static. And the static one basically refers to a script that you would execute in a context, for example, like RStudio here to derive your analysis. And the live one, and we will open that right now, basically loads an R session in the background of your browser. So you can see there are a lot of things getting downloaded that will be much faster on my end than on your end if you're visiting this for the first time, because there is some caching going on. But really what it does, as long as you're staying on this page, so as long as you don't click on any link here that takes you to a different page, you will have one continuous R session running in the browser, and that will lead you from this jumble of data to nice visualizations and discoveries and insights about your data. So what we are focusing on here, if you go down, are really those code cells that you can see in there. And those code cells, they can be executed. So if we click on run code here, it will execute those code cells, never mind the code that is actually displayed here, but it will execute the code and it will display the result of those codes here. And we will go through certain code cells in this document and explain some of the behavior of LightLogR. And you are very welcome to go in there afterwards to discover all the code cells that we are skipping right now and play around with them, because they are meant to be dissected. So for example, here what we are seeing is a typical pipe in LightLogR. We are starting with a sample data set, and we are piping this data set through the pipe operator into a function called ggdays that has some arguments. And we are piping the output of that function into another function, ggPhotoPeriod, that has another argument. And that is then extended by some common plotting interface that is native to or not native to R, but very common in R, ggplot it is called, to do some coordinate transformation there. And you can play around with this. So if you would like to know what this actually does, you can just remove it. And if you rerun the code and look down here in the graphic, you will find that now the scaling here has slightly changed. And we are not starting with 0, but below 0 here actually. And we can look what this photoperiod function actually does. If we rerun the code, then we will see, okay, that gave us some photoperiod context about our data. We can look at what arguments do. So for example, if we remove this faceting argument, we can see that instead of one facet where everything is in, we get one facet per ID, in this case, participant and environment. The alpha level just fixes the transparency of our coloring. The aesthetic fill basically says I'm filling conditional on the participant or the environment, and the geom ribbon just changes the geom of my plot. So now I have a line plot, and if I remove even that command, I'm ending up in my little data frame here that provides me participant column or an ID column, the date time column, and then a melanopic EDI column. And this allows me to dissect what is happening on screen, and I can start over, meaning that I get the original code, and then I can get the original output back. So it is generally very safe to play around with these code cells. The only thing where you have to take care is if you're assigning a variable. So for example, if we are assigning a variable hello world and run that, then in any code cell of this page, if we go in with test, then we will see hello world because those variables will stay assigned. But other than that, everything is fair game in here, and you can play around with it. And if you get an error message, then do not worry. You can just go in and fix that later on. So that is the basic setup, how this live tutorial works. Because we don't have to take care about installation. We can skip that, but just know there is some information in there how you get LightLogR up and running in your local installation. And we can dive straight into import. And we will start with some considerations on what we are actually importing, because every device outputs a different structure in the CSV file or the text file that you're getting. I brought 3 examples. This one is from the Aclumus device, and what you can see here, it has some metadata, some header in the text file. Before you have the column names, and then you have the actual data starting. So you have basically a rectangular data set, but it has some meta information. But that is not a standard. So taking a different device, for example, the Specky, you will get a very different structure that starts out with the column names at the top, and it also has spectral information. So, for example, 380 nanometer, 385 nanometer, and so on. And then the actual dataset starts. Yet another file is from a device from NanoLambda, which works very much differently. So every 24 and 25 lines or so, a new observation starts. So everything from here until here is one observation, and all of the information is contained in here in a non-rectangular structure. And what we want to have for data analysis is a tidy format, meaning that we have one variable per column and one observation per row. And that is a very good structure to do data analysis with. And LightLogR imports all data from variable devices in a way that you get into this tidy format. And you have to provide very little information on how to do that. We're going to start off with an example file that actually comes packaged with LightLogR. So as you download LightLogR, you already have this data file, and this command just looks where this file sits, and we can see it is somewhere here under user, and this is the actual file. It is zipped. And if we want to import this file, then all we have to do is we have to specify the import function with the device that we want to import from. And we provide the file name, we provide a time zone where these data were collected, and because we need some kind of identifier for the participant that we are importing from, and we don't really want this to be this long file name, we just specify it manually as P1. And if we run this code, we get a very rich information about the import that just happened, starting with how many observations, how many files, how many IDs, what the time zone that we set is, what the span of our observation is, what are the intervals that are contained within our data, and a little overview plot that is not very useful right now, but you will see them become more useful as we import more data from different participants. So without having done anything other than importing, we are already knowing quite a lot about our dataset. It is a very regular dataset, 10-second intervals, spanning about a week. And with very little effort, we can also visualize what that week looks like if we pipe our dataset to ggdays. And we can see it gives us a nice output about this week. We will say more about ggdays later on, but right now it is just a very easy function to display our data. One quick note about this pipe operator. As I said, it just pipes whatever is to the left into the first argument of the right. So if I were to do this, it is exactly the same thing. So all this does is putting the thing on the left into the function. But the upside is it is much more readable, especially if you have a lot of functions, because you can say, okay, I take dataset and put it into ggdays. And as we increase the complexity, that will be very valuable. Now, if we have a different device, then we obviously need something to specify what the different device is. And if you don't know whether LightLogR supports this device, you can just execute the function supported devices, and it will give you a list of the currently supported devices for the import. And there are a lot of different devices in there. We also just added the MyEye device, which will go online with the next update of LightLogR. So if you are working with a device that you don't see on there, just send us a sample file and we'll write a wrapper function that we can add to LightLogR. So if you have a different file, for example, from the Specky, then what you need to do is specify the file name, and everything you do differently here is just specify this different device in the import function. just take care that it is spelled exactly as you see it here in the supported devices. And if you run this code, we will get the exact same overview. In this case, we only have 22 hours of data, but we again can import the data very easily. And obviously, we can also visualize the data very easily. Here we just have 1 day. Now to the more complex data set that you saw above. It works exactly the same way. So you just specify the file, you specify the device in your import function, you run the code, and you get the data. In this case, the data set is very small. It is only 9 minutes long, but it would work exactly the same way if it's like 3 weeks long or 4 weeks long or whatever. Now, if you want to visualize this data set, you can see we run into an error. And why is that? Well, it is because we have a missing aesthetic, and this is on the y-axis. By default, LightLogR uses melanopic EDI for visualizations, for example. And if it doesn't have that, then you have to specify that yourself. So if you want to know what kind of variable is available to you, you can just call the names function on your data set, and it will give you an overview of the available variables. And so you can see there are different things in here, like photopic lux or cyanopic lux, and also melanopic lux, which is not quite the same as melanopic EDI. There is a factor that differentiates those two things, but we can use this one in order to visualize the data. Now, how do we know where to specify this argument or how to specify it? Well, if you were in a normal R session, you would just call up the documentation of this function. But a much nicer way, I think, is calling the documentation web page. So this is the documentation web page, and we are already in the documentation for the ggday function. And it gives you an overview of all the parameters, of all the arguments, and also the defaults for those arguments. And you can see on the y-axis, there are melanopic EDI. And we can change them. And while we are at it, we should also change the label here. And every argument is also explained, so you know exactly what you can put in and what not. So taking that information, we can get back to our code cell and say, okay, we will still want to have ggday, but now on the y-axis, we are specifying melanopic lux and melanopic illuminance. Here, we run this code and we get a display of this data set. Not very useful because it's just 9 minutes long, but you can see the principle here. You can also see that the ggday function that we have here works slightly different from the ggdays function that we called before, because it zooms in to a singular day even more. So if we were to exchange this from ggday to ggdays, you can see this is really about a timeline of days, and ggday is more about those detailed looks. So that is how importing works for different devices, but usually you have a little bit more complexity in that you have data sets from multiple participants. And the nice thing is, if we want to import from multiple participants, we can just do that by specifying all the different file names in one variable. So in our case, in the data folder, there are 3 Eclumus files. Here you can see their names. And if we pipe those file names into the import functions of LightLogR, so we are switching back to the Eclumus specifier, we provide the file names, everything else stays basically the same with one exception. You can see here there is the auto ID argument, and this one gets a pattern. And this is what's called a regular expression. So this jumble of ASCII code is actually meaningful because it specifies that we are interested in the first 3 digits of the file name. I actually don't remember when I've written a regular expression the last time. In my opinion, this is exactly what large language models are made for, you are specifying what the pattern looks like, ask for a regular expression, and it will provide you one. So it is really very easy to generate those. And you get a very quick feedback if it worked or not. So if we run this code, we should see the import for all 3 files, and it will look the same, but slightly more extensive. Here we go. So now we have 3 IDs across 3 files, a lot more observations, spanning 24 days in total. And we do have a lot more different observation intervals. So we are still having 10-second intervals the most, but there are the odd differences in the interval, 11 seconds, 9 seconds. And while it is just one observation that's the culprit here, this really can mess up your data analysis pipelines. And we are also seeing some larger gaps in here, and those are also displayed in the overview plot that we can see here. So every time you see this gray area here, those are prolonged periods where data is missing, but where we would expect data because data was collected here and was collected here. So why is there a gap in there? And you have to deal with that in a principled fashion. So you can already see that the overview function is much more sensible here because it relates those different participant measurements with one another. If you visualize those data, again with ggdays, it will automatically create a facet for each of the participants. But you can see this ugly gap in here because it doesn't really know how to deal with that for the function. It is not aware about this gap, and we have to make it aware, which we will do in a little bit. For right now, I'm just interested in removing these days so that we can talk a little bit about visualizations. So I will call this function here, which is called remove partial data. And what it does is it will remove days that have less than 20% of data left. So if we execute this function and get a new variable called dataset red for reduced, we can see that now we just threw away those days that had too little data. Because then we can go on to talk about a little bit about different visualization options. Before we do that, 2 things. One thing is we are going to select a subset of the variables that are connected in the dataset, because for some calculations that reduces the calculation time. So we are just selecting the ID, date, time, some actimetry variables, some light variables. And if we do that, we can see that we end up with a slightly reduced data set. That is the first thing. And the second thing we are doing at the end here is to specify how we're even going to get the correct time zones. So we specified Europe, Berlin above, but there are many different time zones, and it's all contained in this function also names. So if you go in there and you find your exact time zone that you're interested in, that will work with light log R. So you can run this code multiple times and it will just give you a sample, and you can see the basic principle behind those time zone identifiers readily. So most of them are really just continent and then a city that is at the center of the time zone. So that's it for import. We have now imported data from 3 participants. And while we still have to do some validation and gap cleaning, I want to go into some different ways to visualize these data. You already saw ggdays and ggday, but just to highlight that it really is customizable. So there are a lot of functionalities in there where you can change how these functions work. For example, making a conditional coloring. Now you do have redundant information in this plot because you have the ID here and also in the color legend. But if you remove the faceting, for example, from this plot, then suddenly the coloring might make sense again. What we can see here is this doesn't look quite right because those live on different time frames, you know? As we've seen before under the overview plot, this really were collected much longer than, much more before the 218 and 219 participants. So having all of this in one plot in this case doesn't make a lot of sense, but as you add the faceting back in, it it cleans everything up very nicely, which is a very nice thing with the ggdays function. All of the functions in LightLog R for visualization also have the option for an interactive mode. And what this will do is two things. First of all, it will make the figures look like crap, so you cannot use them anymore for any kind of publication, but also you can just mouse over and get where you are in, what is the current value, what is the time of that value. So for explorative tasks, this can be very useful, and you can zoom in and have different sections and snippets of your data. So that might come in handy depending on where you are in your exploration stage. ggday, as we said before, focuses more on individual days. So if we run this code, which contains the basic function with some arguments and then just some general plotting sections, we can see that it gives us those three facets for the different IDs, and then 1 day facet for each weekday. So Monday, Tuesday, Wednesday, and so on. So in this case, it spliced together those two half Mondays because all of them live in the same Monday identifier. And this is basically what we specified in here. If we were to remove that, we would get individual dates and also have like this large distance between the different IDs again. So this is a much nicer way to look at individual days. If you want to really look at long streams of days, you can do that with the overview function. It is called already during import, but you can call it separately. You can adjust and customize it, same as the other functions, because it is really just a plot that is generated with the very popular ggplot2 package in R. So you can adjust it to your liking. So here we just added the coloring. And GG overview is also very useful if you're having longitudinal data. So if you want to see how those different time frames of measurements between participants, how they measure up, you can do that very nicely in GG overview as well. And all you have to do is group the data by the different collection periods, and we will see how this grouping happens later on. GG heat map is exactly that. It gives you a heat map of your data across time. So it is very nice to see how patterns change across time for each ID again. And we have the same basically logarithmic scaling that also contains the 0 lux values, which are very common for light exposure data over 24 hours. You can change or adjust the binning. I won't go into detail about the others here. Just two things I want to highlight. Is one, it has a useful double plot functionality. So plotting 2 days right next to each other before you have so you have day 1 and day 2, day 2 and day 3, day 3 and day 4, and so on. So you can look at those patterns across midnight very nicely. And you can adjust this type of functionality very easily as well to, for example, give you a black and white plot about a state. So if you want to know when a certain state happened, for example, sleep or wake, or in this case, when melanopic EDI was below 10 lux, you can also do that very easily and very powerfully visualize your data. As I said before, we are focusing on light variables for today, but LightLog R very much works with any kind of variable. So if you provide, for example, an actimetry variable PIM, you can see it displays that just as nicely. You have to specify what label you want to have. And obviously it makes sense to think about the scaling of the y-axis because it might not be sensible anymore to use a logarithmic scaling depending on what the variable is. And in the documentation, you can find the different ways how to do that. So those are some basic visualization techniques, and we'll see one or two more later on. But now I really want to go into the validation. And for that, we go back to our original data set. And I want to talk a bit about terminology of gaps in LightLogR. Because where we want to be are in this area here, so the regular time series, meaning that from the first observation until the last observation, we have a regularly spaced sequence of observations, and this sequence is not broken. So even if we do have gaps in the data, which are in some cases unavoidable, then we still want to have some kind of identifier in there that the observation is not available. So those are good time series to work with, because then the functions that we are using our data on are aware about these gaps in the data, and you have to explicitly state how you want to process them further. If you don't have that, so if you're living in this world here of irregular time series, then these different observation intervals really can mess up your processing results, because the data set is not aware that there should be observations in here. And this is what we are calling implicit gaps, and this is an explicit gap. So we want to get from implicit gaps to explicit gaps, and also we want to avoid irregular data, which is simply a data point on observation that does not fall onto this regular interval. And this regular interval that is the most common in a data set is what we are calling a dominant epoch. So those are just some terminology phrases that you will see over and over in the documentation of LightLogR, just so that you're aware how it works in there. If you want to quickly check whether your data set has implicit gaps or has irregular data, there are two functions for that. And in our case, we do have both implicit gaps and irregular data, and we can visualize where those lie with the gggaps function, which is another visualization function. And we specify that we want to get this information by day, so not for the whole participant, but we want to get it for all the days that are affected, and we also want to see our irregular data. And because that can take some time, in this case, we would have reached the limit for execution time, which is 30 seconds. So it doesn't take 30 seconds to execute it in a local environment, more like 10 seconds. But the online version is not quite as fast. So here we just have the output that would be produced by this function, which is this by day plot of where we have gaps, which are the red shaded areas, and where we have irregular data, which are the dots here. And this is very typical for a data collection, because very often you don't collect a whole day's worth of data. So you start somewhere during the day, you end somewhere during the day, and you're left with these chunks of the day that just don't have data. That is unavoidable. What is also very common is that you are collecting a little bit of pilot data beforehand. So you check whether your equipment actually works. So this is what we are seeing here. There is some data collection going on that is very regular, and then the collection is stopped. And when the participant comes in, the recording is started again, but it is slightly off from the regular interval that you had before. So the observations themselves, one to the next, are still 10 seconds, but in here there is this shift in timing, and that leads to irregular data according to the principles that we saw above. And this is basically the very same thing, it's just on a different day. So there is some non-deleted pilot recording on a different day, and obviously that is also something that we would like to avoid. There are different ways how to deal with these kinds of pilot data, but we have highlighted 3 ways how to deal with them, how to remove them. And I will only go through the best of them that I consider the best of them. You can try all the others for yourself afterwards. There are different ways, but I think the best way is also the most principled way, which is that you define the actual trial times. So when did a participant wear a device and when did they take it off? Usually that is stored in a CSV file. Here we have to reconstruct a variable that contains that. Let me just show you what that looks like. So all of this code really just creates a small data frame that contains the participant ID, start and end time, and says, okay, this is the trial time. Yeah. And we can use that to add it to our data set. So there is a lightlogr function called add states, and it uses data sets that are shaped like this, and it will add this information to our data. So what does that look like? We can show you that. So if we run just this code, so we pipe the data set to add states that has been given the trial times, then it will give you the data set back with an additional variable called trial, because that is what we specified here. And every time this is not the case, any of these rows, then it will just give you an NA. And whenever there is actually within this trial times, then it will state true, and we can filter by that condition, which is what's happening here. So we add this information to our data, we filter by this information, and then we remove that trial column because we don't need it anymore. And if we do that, then we can see there are still gaps in the data, but those will be just the chunks of the day where data was not collected. So we only end up with 6 days where no data was collected, and then we have nice regular data until the end of the collection episode. And this is just very common, so that is not really problematic from an irregular data perspective. If you want to know from a more numeric perspective how your data are regarding to gaps, then there is the gap table function, because that is also a little bit problematic in terms of the WebR environment that we are in. I'm also just showing the output here, but it gives you this nice table where for every ID and overall you're given a number about what is the regular data that you're having, what is irregular data, what are the gaps in your data, and is that gap implicit or explicit. And for our data set, we can see about 13% of our data are implicit. So those are exactly those times here. And what we want to do is make this data explicit so that all the functions that we use in lightlogr know about this explicit gap. And we can very easily do that with the gap handler function. So what this function does is it creates this regular time series. It fills all the missing observations up with an NA. And if we set the full days true argument, then it will also look for the first and the last portion after the edge observations. And if we execute the gap table on this data set, then we can see we get exactly the same information, but now we do have 13% explicitly missing data. And that is exactly where we want to start off with our more numeric analysis. If we now run our functions again, we can see that we don't have implicit gaps anymore. We don't have irregular data anymore. And if we visualize our data set, we can see that again, we removed these problematic time zones before, or time observations before noon, basically on the first Monday, and afternoon on the last Monday. And now it's time to decide which days we want to keep. Right here, it is pretty easy to decide that probably we don't want to keep the Mondays in here, but usually if you have longer gaps in your data set, that might not be the case, that it's just about the first and the last day. And we come back to this function, remove partial data. So remove partial data basically looks at groups of your data and says how much data is missing in this group. And if it is above a threshold, it will remove that group from your data. So in this case, we are looking for missing data for melanopic EDI. we are specifying that we are interested in days that have no more than 1 hour missing, and we want to have this filtering done by day. So not for the whole participant, but for each day of each participant. And if we run this code, what we will see is that the dataset now starts on Tuesday and ends on Sunday because those edge cases did not fit this condition. And again, it is pretty simple to see in this example, but it is very important to go through this in data that has more gaps, and that is very common in many different data sets, which is what we describe here. On to metrics. There are a ton of metric functions contained in LightLogR. As I said, we are not interested in telling you which functions you can or cannot use. We are interested in giving you a consistent interface to those functions. So there is a lot of documentation on the web page for LightLogR on how these functions work and when to use them, how the different arguments work. And I want to show you how to access these functions in LightLogR using the, where do we have it? Duration above threshold. So basically, this is what in the literature is often called time above threshold, time above 250 lux, for example. So we want to showcase that one and also the interdaily stability function. So we'll look at those specifically. The principle here really is that you call the function and you provide a vector for a variable and you provide a vector for the time. It says light variable, but again, it is not limited to light. You can provide any kind of other variable in there and will just as well do the calculation. It is just created with light variables in mind. So if we want to have time above 250 lux metanopic EDI, we specify a threshold of 250 lux. We provide the metanopic EDI variable and the daytime variable. And if we run this, it will give us an output of 2.1 days roughly that we are above threshold here. And this really is for the whole vector that we are providing. We can check the other way around as well. So there is an argument in there that allows us to specify that we want to get the time below 250 lux. And if we execute that, we can see it is a much longer period, obviously, over 2 weeks. And so this is how you, in principle, calculate these metrics, but it is not a very useful way because you are doing it for the whole vector. So we are talking all measurement times and all participants. Right here, this might still be useful in some cases, but it gets very much useless if you're looking at, for example, the brightest 10 hours of the day. You can execute this function, which will give you by default the brightest 10 hours. You can also specify the darkest 5 or whatever. But if you provide the whole data set, it will not be a useful output because it will just look at the brightest 10 hours of the whole data set. And especially here where you have overlapping data, that can be problematic. So you need a more principled fashion on how to access these data for different subsets of your data set. And the way to do that is with the summarize function. So you pipe your data that has some grouping. Let me show you. If we run this data, we can see there are groups in there. And it is by default, LightLogR groups by ID upon import. So you always group per participant, 3 participants, so you end up with 3 groups. And we pipe that into summarize. We again specify our metric function the same way. We just don't have to reference the dataset anymore because it is already aware of that. And we also have to specify as a data frame. This allows automatic naming of the output variables. And if we execute this command, we can see we get a nice table that gives us 1 result per group. So 1 ID, 1 participant, 1 result. And if we want to have more than 1 metric, then we can just add to that. So here we have duration above threshold, we have interdaily stability, we provide everything within the summarize command. And if we execute that, we can see that, again, we get a very nice table containing one outcome for participant 216. It has 1.03 days above 250 lux melanopic EDI and an interdaily stability of 0.26. And you can add as many different variables as you would like to that roster. Can be 50, whatever. It will still give you those pieces of information. The thing is, sometimes that is not granular enough. For example, here for duration above 250 lux melanopic I, usually you're interested what that is per day and not per participant. And the way you would do that is by grouping the data additionally by date, not only by participant. You can do that manually, actually, if you have a variable, or there are convenience functions in LightLogR. For example, here, add a date column, group by that column, and I don't want to have the date, I want to have the weekday. And if we execute only this portion, then we can see that what we are getting is our data set, and we just have an additional column in there that says Tuesday or Wednesday or whatever else the current date actually is. And if we pipe this extended data set into our summarize function, then what we will get is an extended set of outcomes here for each participant. On each day of the week, we get the information about 250 lux melanopic EDI or higher, 4.27 hours for this participant on a Tuesday, and about 30 minutes for this participant on a Tuesday. And then we can use some kind of summarize functions. There is a convenience function again to summarize numeric values down. And so we know, okay, participant 216 has about 4 hours of time above threshold on average for each day, whereas we have 1.4 and 2.8 hours for the other participants. And we have 6 days that factor into this average here. So we are getting very quick insights into the different metrics for these participants if we group them in this structured fashion. So that is how you calculate metric. Now I want to dive into 2 very small sections that are very important, I think. One is photo period, because photo period is just such a powerful way to structure your data that requires very little. Actually, all you require are coordinates. So if you know about the coordinates of your data collection, you can extract when did the sun go down, when did the sun go up, what was the photo period in hours, when was it daytime, when was it nighttime. So you can obviously extract that information, but you can also add that information to your data set. Just specify the coordinates, and you can see you get 4 new variables in your data set about dawn, dusk, and photo period, and also the state of the photo period. So is it day or is it night on each of the observations? And you can use that information for metric calculations or for visualizations. There is a convenience function called ggPhotoPeriod that we came across in the very beginning. It just adds on to one of the visualization functions, ggday or ggdays, and it will just shade by your photo period. Very nice and easy. And it will work either by looking for photo period information that is already in your data set, or if it's not, which is the case in our reduced data set that we generated before, we can just specify those coordinates in the photo period function itself. So it's a very nice and easy way. And you can see those are 2 different data sets, because here we remove the Mondays, and here the Mondays are still part of the data here. So you can also do this or add this photo period information in order to structure your data and group it for daytime and nighttime metrics. I'm not going to go through the pipe that we have here, but basically what it does is what we've seen above, but additionally it looks at daytime and nighttime conditions. So you know this person at night never had values above 250 lux melanopic EDI, where there were some minutes for the other participants on average. So photoperiod, very easy and very powerful, I believe, for analysis. And the other thing I want to quickly go into is the distribution of light exposure, which is something you have to keep in mind when you analyze these data, because participant data, so personal light exposure, if it's collected through a long period of time across 24 hours, usually contains some zero lux values, especially at nighttime, but also during daytimes. And those are not always non-vare times that you would actually have to filter out. Sometimes this is just the case during daytime as well. And very often in publications, I do see the average or the mean for certain days calculated, and I just want to showcase that that is not a very useful metric. So if we calculate for our data the mean, the median, and the geometric mean, we can see those values are very much different, and they live on different scales even. And if we visualize these different statistics for our data set atop the distribution of the data, you will quickly see that the mean, which is this red line here, is not a very useful descriptor for any kind of light exposure metric. So it is much too high. This is the geometric mean, which usually is a very good descriptor of the non-zero part of your data. And the median here, as you can see, factors in the zero lux values of your data set. There is a way to keep within this mean or average framework, because the median sometimes is not as statistically, how would I say, so you wouldn't want to work with the median because it is, for example, not contained in a lot of statistical inference functions, which work on an average basis. So if you want to stay in that context, there is a function called log zero inflated, which adds a small portion to your data set before it logarithmically transforms your data. And obviously you can back transform it with another function. And if you calculate that, you are getting something that is much closer to the median in all cases. Usually it's a little bit below that, and very much represents this duality of your data between a logarithmic distribution and the zero lux inflated values that you're having. So here is exactly the same graphic that we've seen before, but here the lower one is the log zero inflated value and the higher one is the median. So working in statistical inference with some kind of averaging, I would suggest transforming your data with this type of convenience function. And it is very, very easy to decipher when you've used it. So for example, if you look at your code, then you just add this functionality on top of your actual variable that you're providing, and then it is very clear for a person looking at your code that this type of transformation has happened. And it really does make a difference. So if we are looking at average values of personal light exposure for those participants, then we can see the untransformed average is 550 lux for this participant, and it's about 10 lux if we factor in all those zero lux values on the logarithmic scaling that we had here, which is probably a much better descriptor for where our light exposure data lie. So two important considerations here. There are two summary functions that we are just about to add to LightLogR. The first is called grand overview, and the second is a table summary. And those, I think, will be very popular functions because with very little inputs, they provide a very nice summary of your data. So where was it collected? When was it collected? What was the photo period during the collection like? And also what is the typical pattern of light exposure that we are seeing here. So you can't execute that in the virtual environment here, but you can do it if you work with the static version. So just to prove that this is a real function and not something I'm faking here, you can execute that if you execute the standard version of the script, and you will get this output, and it will be contained in the next version of LightLog R. Same thing for the summary table, which I think is very nice because it gives you a little bit of metric overview of your data, how many participant days, what is the missingness of your data, what is the photo period like, and then just a whole slew of typically calculated light exposure metrics like mean timing above 250 lux and so on. So those will be very useful overview functions for many people out there, and I expect that this is also a very nice way how to summarize things for reports or publications. So let me use the last minutes of our live session to highlight a few processing functions that are very important and that I really constantly use. The first one is aggregate date time, and this really is a great function because this one allows you to resample your data to whatever interval you desire. So if you want to work with hourly data, you specify you want to have hourly data. If you want to work with 15-minute data, you specify you want to have 15-minute data. The way it works is it bins all the data points within the units you're specifying and then applies some kind of function. And it has default handlers for all the different data types. So if it's a logical value or a character value, it will just give you the most often used within these timeframes. It is the average for a numeric value. And you can change all of these functions, these handler functions to your desire, so you get the output that you want. And this is really a great way to change the interval of your data, because usually you're starting off very small, 10 seconds or something like that, because you want to know about the variance of the data, and then you're usually more satisfied with something in the 5 or 15-minute range, because let's face it, if you have a lot of data from many participants, calculations can get quite slow. So changing the unit, the interval of your data really is a very nice way how to handle that. And also there are much worse ways to handle irregular data than to set a different aggregate of the time. So if you have slightly irregular data at the 10-second level, then aggregating it to 5-minute values really deals nicely with the irregularities if they are not too large there. Another function that builds off aggregate date time is aggregate date. And what that does is it will condense everything within a group down to a singular day. So if you specify 3 groups, 3 participants, then you will get a single day per participant. And you can also resample the interval here. And this is really very nice if you're dealing with weeks of data and you would just want to have an overview of what is the typical light exposure from this participant like. And the only problem that we have here is now we don't really know how this pattern works across midnight. But here comes ggdoubleplot to the rescue. It is a function that just plots 2 days right next to each other. So a little bit similar to the heat map functionality, but in this timeline plot here. And you can see we can see the patterns across midnight very nicely. So these are still average days for these participants in a 30-minute interval and plotted as a double plot, so 2 times the same day. And obviously we can enhance this visualization further by adding some photo period information to that, which we will do in the next code cell. And that really gives us some very nice interpretability about what these data looked like. So with very few lines of codes, very few pipes that we can just go through one by one, we arrive at a very, very powerful visualization. The nice thing about aggregate date is that it allows us to specify additional values. So for example, if we not only want to know what the average timeline was for this person, but we want to know what was the minimum and maximum version, we can specify those variables as well. And we can plot them just through standard ggplot interfaces here. So if we run this piece of code, it will give us the 30-minute interval for each participant, including the minimum maximum value for each time point, which is, I think, a very, very nice way to showcase the variance in there. And you can obviously do that with certain quartiles of the data if you don't want to look at minimum or maximum. So it's really up to you to specify that. So that's, I think, as far as I want to take it for this live session. There's a little bit more to come. So in your own time, go over those things, explore them. We tried to really annotate all these different functions so you know what you're doing. And if you go through this in your own time, we think you will discover many different ways how to interact with your data, to annotate your data, to process your data. And hopefully this has given you the kickstart that you need to interact with LightLogR. There are many more things that we haven't been able to talk about, but there are more courses to come, and hopefully this has given you a little impression about how to import data, visualize it, a little bit of processing and validation, and obviously metric calculation. So with that, I would like to end the live session and go into the Q&A session. And let's see what kind of questions you come up with.